Namaskaram, Namaste, good evening to good afternoon to one and all present over here. There are some participants from Ghana as well. So for them, I would like to say Memawaha because it's afternoon, I think, over there. I hope I pronounced it correctly. So a brief introduction about myself. I am Narendra Singh and I'm currently pursuing my MSc in biotechnology from Amity University, Kolkata. I would like to pursue a career in research after this, and my research interests include redox biochemistry of neurodegenerative diseases, with main emphasis on Parkinson's. I'm also interested in studying cancer biology. So this paper actually it's about nitrosative stress-induced apoptosis in dopaminergic neurons and how it leads to neurodegeneration. So next slide, please. Thank you. So in the introductory sentence, I spoke about nitrosative stress. So what exactly is nitrosative stress? So if you look into any paper, you would find the definition as the overproduction of nitrogen-based free radicals, mainly nitric oxide and nitrogen dioxide. So when these free radicals get accumulated in our system, in our body, it leads to a condition known as nitrosative stress, which interacts and dysregulates, or rather I should say that it malfunctions. It's not a very good situation, and it leads to dysregulation of the homeostasis of our body. Why I'm saying this is because nitric oxide and nitrogen dioxide, they are radicals and they have an unbalanced. Yes, thank you. It's Maha. Okay. So these atoms have unbalanced valence electrons, and hence in order to stabilize themselves by filling their outer valence shell, they react with other atoms and molecules and lead to the production of the secondary free radicals such as peroxynitride and hydroxide anion, as well as toxic non-radicals like hydrogen peroxide and dinitrogen dioxide and nitrous acid, which can be very fatal for the cell because these non-radicals are extremely toxic. Next slide, please. Now we find the main culprit in this case, nitric oxide. So from where is this nitric oxide coming from? Well, actually, nitric oxide is not a culprit, and it has some good functions as well. But the problem arises when the production gets exceeded. So nitric oxide is mainly produced via an enzyme known as nitric oxide synthase. And this enzyme has three isoforms: NOS1, NOS2, and NOS3. NOS1 or neuronal NOS, as the name suggests, it's mainly expressed in the neurons. NOS2 or iNOS is mainly expressed in the macrophages. And NOS3 or eNOS is mainly expressed in the endothelial cells. So the numbering 1, 2, and 3 is done on the basis of in the order in which they were discovered. The one which was discovered first was named as nNOS or NOS1, which is mainly expressed in neurons. NOS3 is expressed in the endothelial cells. And there are similarities in NOS1 and NOS3 because they are constitutive in nature. These enzymes require calmodulin and calcium to function as cofactors, but NOS2 is inducible, and hence the name comes iNOS. It is not constitutive. It though generally it's expressed in macrophages, but it can be expressed, the expression can be seen in each and every kind of cells of our body. So the first two examples, as I mentioned, NOS1 and NOS2, they are dependent on the calcium ions, and they produce very low levels of nitric oxide, but INOS produces a very high level of nitric oxide. It is inducible, and it is also not dependent on any cofactors like calcium, though calmodulin is required for its activity. So NOS1, again, this protein is originated, the source is NOS1 gene, and each of the, we will be mainly focusing on the nNOS over here, because we are focusing mainly on the neurons. So each nNOS has an oxygenase and the reductase domain. The name itself suggests that the one would lead to the oxidation and the other would lead to the reduction. So we are mainly playing with electrons over here. And the oxygenase and the reductase domain are connected by a linker, which is responsible for the binding of the calmodulin. Now, calmodulin is an important cofactor in this case, and the generation of nitric oxide occurs in two steps. The first step is that intracellular calcium ion, or Ca2+, it catalyzes the binding of nNOS to the calmodulin, of course, as I mentioned before, and in the presence of heme, it hydroxylates L-arginine, the L form of arginine amino acid, to form N-hydroxy L-arginine. And in this step, the cofactors like FAD and FMN are required, which aid in the transfer of the electrons from the NADPH to the nNOS. So as I mentioned, we are mainly playing with electrons over here. So electron is being transferred from the NADPH to the nNOS to the heme. And in the second step, now since the electron has been transferred, the N-hydroxy L-arginine, it's oxidized to form L-citrulline, and nitric oxide is released. And again, this is done with the help of L-arginine and a cofactor tetrahydrogen borate. Next slide, please. Now, coming to the disease which we are targeting at, Parkinson's disease. Now, Parkinson's disease is caused by loss of nerve cells in the part of the brain called as the substantia nigra. Now, these neurons mainly help. They mainly are dopaminergic neurons, as they are rich in dopamine, and they have a lot of dopamine receptors over there. Nerve cells in these, now, what is the role of dopamine, first of all? Dopamine acts as a messenger between the parts of the brain and the nervous system, which helps in controlling and coordinating the body movements. Now, if these nerve cells die or become damaged, the amount of dopamine in the brain is reduced, of course. And due to this reason, the part of the brain which controls the movement cannot work as well as normal movements, which causes the movements to become slow and abnormal. The loss of nerve cells is a very slow process, and the symptoms of Parkinson's disease starts the symptoms, they actually start when 80% of the nerve cells have been lost. So Dr. Dahel is saying something. Yes, absolutely, Dr. Dahel. Now, as I have also mentioned in the slide, that when 80% of these cells die, it leads to the onset of the Parkinson's disease. 80% means there is almost total depletion of the dopamine containing neurons. Next slide, please. Now, the question again is that how do nitrosative stress results in the loss of dopaminergic neurons? I have spoken about the main culprit over here, which is nitric oxide, but actually nitric oxide is beneficial in most of the cases. It starts the body starts malfunctioning when the nitric oxide production increases by a large extent. Now, again, INOS, as I mentioned, it produces a very huge amount of nitric oxide in our body, and which is the inducible nitric oxide synthase, and it is expressed by the excuse me, it is expressed by the glial cells such as astrocytes and the macrophages in our body, in the neurons. INOS, again, the structure details have been given. What causes nitrosative stress? Again, Dr. Dahal, as I mentioned, excess production of free radicals such as nitric oxide and NO2. So when they are in excess quantity in our body, it leads to nitrosative stress. Now, INOS, again, the details have been given that the enzyme is a protein which is of 131 kilodaltons in size, and it is encoded by NOS2 gene. The main details which I would like to show, it's not oxygen metabolism disorder, doctor, it's a nitrogen metabolism. Not exactly metabolism, actually, it's the production of free radicals. So again, because as I've also mentioned before that the expression of NOS in neurons and the ENOS, yes, nitric oxide and the superoxide. Actually, when nitric oxide is produced, it is highly unstable and it reacts with other atoms and molecules. It also forms ONO minus, which is, like as I've mentioned before, it's peroxynitrite. So of course, it's an interaction between nitric oxide and the oxygen. But again, it can be due to any particular metabolism and not particularly. Yes, definitely, they are the free radicals, and not particularly, the oxygen metabolism. It can be because of due to any metabolism from in our body. Now again, the constitutive expression of NOS in the neurons and ENOS as well, but INOS is inducible, and hence INOS binds to calmodulin at a much lower concentration of calcium. As I mentioned, calcium is an important cofactor in the case of NOS, but not in the case of INOS. And it produces an tremendous amounts of nitric oxide. Now, elevated levels of INOS have been found in the post-mortem in multiple neurodegenerative diseases, including the Parkinson's disease, which means that again, since the INOS at a very low, doctor, oxidative stress is mainly caused due to the production of free radicals of oxygen, derivatives of oxygen, and a nitrosative stress means it must have an element of nitrogen in this case. That means, of course, oxides will be there, RNS will be there, which is the reactive nitrogen species. But in case of oxidative stress, it can be oxides of any particular elements in our body. But in case of nitrosative stress, it has to be specifically nitrogen. So again, the elevated levels of INOS have been found in the post-mortem in multiple neurodegenerative diseases, including the Parkinson's patient's brain. Again, the glial cells are they are present in a very high population in the brain region, which undergoes active neurodegeneration. That means the glial cells are targeted very much when these free radicals are present in the very large concentration, which increases the potential for high nitric oxide levels that can further exacerbate toxicity in the surrounding cells. Next slide, please. Now, as I mentioned about nitric oxide, now the question again is, how do nitric oxide mediate changes in the axodendritic functions? So as I mentioned, nitric oxide production is good for our body, but again, when it goes to excess, when the production is in excess, it leads to the nitrosative stress, which is very detrimental. Now, NOS contains a particular domain, which is known as the PDZ domain, which confers the binding capacity to many postsynaptic density proteins and is therefore regionally distributed along synaptic spines. Now, the localization is the key if the NOS has to express itself, because when it is distributed along the synapse, it produces the nitric oxide signaling, the second as a second messenger signaling. The first messenger signaling in the neurons is, of course, we know of that of the calcium and the sodium potassium ion channels. In this way, nitric oxide reacts with guanylyl cyclase to produce cyclic guanosine monophosphate, which is again important for regulating the dynamics of the neurons. This includes the outgrowth and synaptic plasticity. In addition, nitric oxide has a major role in regulating the synaptic activity through S-nitrosylation or trans-nitrosylation of the proteins. In a healthy state, like in the normal state, nitric oxide protects the neurons from the hyperexcitability by S-nitrosylating and inhibiting NMDARs. Next slide. In the next slide, I'll be explaining this in a bit more details, how it can be detrimental. Next slide, please. Again, nitric oxide has also been shown to protect neurons from hyperexcitability by regulating the AMPAR expression, which is a major component of ionotrophic glutamate signaling. AMPAR surface expression is increased or decreased in the presence of SNO-stargazine or SNO-thorase in response to the S-nitrosylation of respective regulatory proteins. In case of neurons, nitric oxide itself acts as a neurotransmitter. Why I'm saying so? It's because of the calcium signaling, which, as I mentioned, it's an important cofactor, and it stimulates the extracellular vesicles to release an endocytosis, and is even involved with learning and memory formation through its activation of the CREB and the retrograde transmission from the post to presynaptic terminal, stimulating the neurotransmitter release and long-term potentiation. So this is the pros and the cons. Now, mainly coming to the cons part, when in excess, as in case of the Parkinson's disease neurons, nitric oxide alters the axodendritic function altogether, which impairs the synaptic signaling, vesicular trafficking, and dopamine homeostasis. It dysregulates the homeostasis of the dopamine, as I mentioned before. It also traffics the vesicular networking and the synaptic signaling. That means it alters with the signaling pathways altogether, which can be fatal as well. Next slide, please. Now, how do nitric oxide induces the instability of the mitochondria of the dopaminergic neurons? Now, as we all know, mitochondria produces ATP. It's the powerhouse of the cell. And when the demand for ATP is heightened, like in the case of the synapse, nitric oxide regulates the oxygen consumption of the cell. Speaking in layman terms, nitric oxide actually helps in regulating the respiration of the mitochondria. Now, again, this regulation is important in context of the substantia nigra dopaminergic neurons, the part which is mostly affected in the case of Parkinson's disease, given their complex arborization and pacemaking activity. Also, the substantia nigra dopaminergic neurons have very fast spiking CO2 transmitters, transients, that increase the intracellular concentration of the Ca2 plus ions. Now, Ca2 plus acts as a cofactor for the NOS activity, and this expression is usually buffered by the mitochondria. Now, this puts mitochondria at a risk because of the burden of the Ca2 plus ions present, which can also lead to excitotoxicity or apoptosis. But Ca2 plus that is not buffered, suppose if Ca2 plus is not being buffered by the mitochondria, in that case, NOS gets further stimulated to generate nitric oxide. Also, mitochondrial nitric oxide reacts with superoxide produced by the mitochondria. As I was also explaining before, that this superoxide, the nitric oxide reacts with the superoxide to form peroxynitrite, which is extremely toxic for any kind of cell. Now, these peroxynitrite not only impairs the mitochondrial ATP synthesis, but also it inhibits the polarization of the mitochondria and detoxifying enzymes, which again adds to the promoting mitophagy. That means the autophagy of the mitochondria. It's just a fancy term. Next slide, please. Now, how reactive nitrogen species can be used in the therapy of Parkinson's disease? We have spoken about the problem, and now coming to the solutions part. There are mainly two ways. Broadly, there are two ways which are present right now. So if we are to kill the culprit, we can kill its ancestors altogether. So that's what we are doing in the first case. We are reducing the reactive nitrogen species by directly targeting the nitric oxide synthase enzyme. So in this case, we are upregulating certain proteins which can inhibit the neuronal nitric oxide synthase production altogether, which means that mitigating Parkinson's disease pathogenesis. Now, the specific NOS inhibitor 7-nitroindazole protects against the dopaminergic neuron depletion in animals treated with 6-OHD8-126 or MPTP12. Now, it also prevents the motor impairments such as apomorphine-induced contralateral rotations and catalepsy. N-nitric oxide synthase inhibition by N-nitro-L-arginine methylester can also reduce nitric oxide accumulation in human-induced pluripotent stem cells, derived Parkinson disease neurons, which protects the neurons from mitochondrial dysfunction and rescues axodendritic pathology. Also, treatment with docosahexaenoic acid causes the phosphorylation of the NOS. That means we are adding a phosphate moiety on the enzyme, which decreases the NOS activity. That means it is inhibiting the activity of the neuronal nitric oxide synthase and prevents Parkinson disease-like motor deficits in MPTP treatment. And this has been seen in mice. When we are inhibiting nitric oxide synthase, we can also do one thing, which is nitric oxide inhibition can be regulated by the addition, by the alteration of the gene, by silencing the gene by using the interfering RNA. This could also degrade RNAs that are integral for nitric oxide synthase synthesis as well. iRNAs, or the interfering RNAs, are delivered to the cells through viral or nanoparticle or liposomes or non-viral vectors, which can be adeno-associated virus or retroviruses. Adeno-associated viruses or retroviruses in case of viral vectors, and in case of non-viral vectors, we can use the nanoparticles or liposomes. There is a mistake in the slide. Kindly check that out. Now, similar to the protection from the Parkinson disease phenotypes exhibited by the neuronal nitric oxide synthase knockout mouse, striatal inoculation of the interfering RNA targeting N-nitric oxide synthase reduced 6-OHDA toxicity can be maintained by preventing the dopaminergic neuron degeneration and behavioral impairment in rats. The clinical trials are being ongoing, which employs the use of interfering RNAs for Parkinson's disease. We haven't reached much success till now, but we can definitely do so in the coming decade or in the coming years. Next slide, please. Now, again, the second method. In the first method, we were mainly targeting the ancestry of the nitric oxide, which is the enzyme nitric oxide synthase. In the second method, we can be using some kind of antioxidants like denitrosylating proteins. Because of the nitrosylation, we have all seen that it leads to the nitrosative stress. So, what if we target the cells with proteins which are denitrosylated? Since the nitrosylation of proteins has deleterious effects with immediate implications for Parkinson's disease, targeting this modification may offer therapeutic benefit. For example, S-nitrosylation of the peroxy reductase has been shown to impair the hydrogen peroxide reductase capacity of the PRX2, resulting in increased oxidative stress. Now, again, the S-nitrosylated and the PRX2, when they form a complex together, it is found that the Parkinson disease models, these complexes are in more abundant than controls. Again, the most well-studied protein for nitrosylation or denitrosylated function in Parkinson's is parkin. The oxidation of parkin leads to its aggregation, which reduces the ability to act as a redox sponge. Now, it has been reported recently that the protein DJ1 regulates SNO-parkin and the S-nitrosylation of the other proteins via the trans-nitrosylation. Now, this inactivation of DJ1 mimics the Parkinson's disease-related mitochondrial dysfunction, and besides regulating the SNO-parkin, SNO-DJ1 itself also represents a potential Parkinson disease therapeutic target, since DJ1 plays a role in activating the antioxidant response, cellular metabolism, cell survival, and redox homeostasis. However, the specific function of SNO-DJ1 against the denitrosylated DJ1 have yet to be elucidated. Further research that identifies supplementary denitrosylating proteins may provide valuable information for the removal of SNO modification and rescuing the Parkinson disease pathology. Next slide. Now again, thank you very much for your patient listening, and any questions from anyone, I'll be very more than welcome. Thank you.